Hi, today I'm going to talk about sparse signal processing theory and its application to bioinformatics and computational biology. I will mainly talk about gene expression reconstruction and also introduce some of my work related to this topic. Well, suppose that I want to know what happens in my cell in detail. Then I need to investigate gene expression of the cell. High-throughput experiments such as microarray or RNA sequencing are nice in terms of resolution and coverage, but it's still too costly to perform daily experiments. Although qPCR or real-time quantitative PCR is accurate and cheap, but it's not well scaled. If I have a method that can generate high coverage expression profile with reasonable resolution and feasible cost, even though I may lose some precision, I can understand the entire system of my cell, as a microarray or RNA sequencing does. So how is possible? So let me introduce some possible method later. In 2017, a research group in Broad Institute proposed compressed measurement of gene expression. I'll tell you what this statement means in a few minutes, but let me introduce this concept first. Suppose that you have a large amount of data collected and want to compress it. Assume that the data is an image and the compression algorithm is JPEG. As you may experienced before, when you store image in JPEG format, you lose a little bit of resolution, but it is smaller and still recognizable. But think about this. If your primary objective is just want to get the recognizable JPEG image, why do we do this inefficient job? Instead of collecting large amount of data that can be a very high resolution of image, can we measure a small amount of data and recover or approximate JPEG image by data decompression algorithm? This is the same problem that I introduced in the previous slide, and the decompression algorithm is called compressive sensing algorithm. So how they actually work? What JPEG does is that it performs Fourier or wavelet transform and make all coefficients 0 except 1 or 2% of large coefficients. When it back transforms this few non-zero value containing matrix, then we can get exactly the same image. The compressive sensing does exactly the opposite. It randomly samples pixel values, let's say 10%, and solve the constraint optimization problem. Then the objective function of this problem is minimizing error of reconstruction while minimizing non-zero Fourier or wavelet coefficient, as we have seen in the JPEG example. In other words, the problem is to find sparsest vector S, which gives you the minimum reconstruction error. Let me try to explain this principle with a little bit of math to fully grasp this idea. Let's say Y is a low-dimensional composite measurement, and S is the sparse coefficient, as you have seen in the previous slide. The job is finding the sparse coefficient vector without compression. By the way, this vector is just vectorized image, meaning that concatenation of matrix. Instead of solving left form of equation, see right side of equation. It is a familiar y equal ax linear algebra problem, and you can solve it with x equal inverse ay. And actually, this is not possible because you have more column than row. Recall that we can solve this type of problem if we have equal number of equation and variable. Therefore, this is not a simple linear algebraic problem, but constraint optimization problem. And the constraint is the number of non-zero element. By minimizing non-zero element, we can find optimal solution of this problem. And thanks to the great mathematicians like Donohue, Candace, and Romberg, and Tao, we now know that this problem is equivalent to the minimizing L1 norm of my sparse vector. The exact mathematical form of objective function is shown in Lagrangian format, and it is just weighted average of reconstruction error and L1 norm of non-zero coefficient. So you want to minimize the error as well as number of non-zero elements at the same time, and the importance of two term is weighted by lambda. You may not familiar with L1 or L2 norm. I will explain it briefly. For 2D case, the L2 norm is also known as Euclidean norm, and it can be represented as unit circle. And the L1 norm is just sum of magnitude of vector x and can be represented as a diamond. L not pseudonorm is just plus sign denoted as yellow bar in the picture. If I have a bunch of data points along with the line as in the picture, the solution of L2 norm has both x and y components because the intersection contains x and y coordinate at the same time with high probability. L not pseudonorm form either have x or y component, and L1 norm is very likely have only x or y component because solution space of the L1 norm forms a diamond. In high-dimensional space, this property still holds true, though the way of intersection between L1 hypercube and my data points may differ. You can think this whole discussion in terms of linear regression. In multiple linear regression, you want to find best linear fit line within the space represented by multiple variables. The L2 constraint promotes equality, so you have similar weights for all variables. In contrast, L1 promotes zeros, so you have most of zero weights for your regression model. In other words, you have fewer variable, simpler model that explain your regression line than without L1 constraint. As we discussed earlier, to perform compressive sensing, we need to know the data is sparse, and the gene expression is modular and sparse because cells do not express genes individually, but it sets in discrete programs, and each cell expresses only a limited number of programs at a time. Based on this observation, in 2017, broad research teams generated random composite gene expression profile from public gene expression database by taking linear combination of genes with random Gaussian weights. Here Y matrix denotes random composite measurement, form a m by n matrix, and random composite weight have a size of m by g dimension matrix. The gene expression vector has gene by number of sample dimensions. Therefore, y equals to ax. As you can see in the figure A, we can construct this composite measurement by a linear combination of expression measurements. There is a decomposition algorithm called blind compressive sensing sparse module activity factorization, also known as BCSMOD, showed good agreement of various sources of gene expression, as in the figure B and C. Figure B shows spearmint-Rankin correlation coefficient between original and reconstructed expression profile of compressive sensing method, denoted here SMOF. With various data sources, say multi-tissues, cancer compendium, and even single-cell expression profiles, the method showed good performance, and the global trends are resembled to the original expression, as in C. Notice that although they use different names such as module dictionary or activity, but it is the same as transform basis psi and k-sparse vector S as we have seen in the compressive sensing theory slide. I also designed one method to solve the same problem using convolutional neural network. Thanks to the recent development of deep learning, we can now solve the equivalent of L1 minimization problem using deep learning. This model is convolutional stacked onto encoder model with skip connections and works in a similar way to how compressive sensing works. It contains encoding and decoding part corresponding to the transform and back transform of data, and by training learns how to minimize reconstruction error in given sparsity condition, let's say 10% of randomly selected data points. By using only 10% of original data, I could achieve 0.86 correlation coefficient value with this model, and this value is similar to the state-of-the-art gene expression reconstruction performance. The fundamental advantage of this model is that it can reconstruct full gene expression from any combination of, let's say, 1,000 genes. Although I explained compressed sensing in terms of gene expression reconstruction, basically sparse signal processing or compressed sensing theory is well developed in the field of bioimaging. Professor Jongchul Yang's lab at KAIST is specialized in this domain, particularly emphasized on the reconstruction of CT, MRI, and ultrasound imaging. Here I attach the entire reference of my presentation. I also made a demo of this material and uploaded it in my GitHub repository. For those who want to study this topic, please refer to these materials. And now I will explain some of the interesting questions related to this topic. The first question is validation of the composite measurement. How can you explain mentally about the composite measurement of gene expression? Suppose we want to know abundance of a single gene. One way to do this is that we design proof for the specific gene tagged with barcode and amplify to measure the barcode through quantitative PCR. Composite measurement can be realized by measuring 2 or more genes simultaneously with a randomly weighted proof mixture. In other words, we put different quantity of proof in a single well. Let's say measure gene 1 with 100 proofs and gene 2 with 50 proofs with the same barcode and perform single PCR reaction and measure the weighted sum abundance of gene 1 and gene 2. Here my ratio is 2 to 1. In the paper, they compared expression of 24 genes in K567 cells with a composite measurement of 8 different random combinations of genes and individual measurement. The expression measured well correlated as shown in the figure D. This type of experiment can be scaled with Luminex or NGS technology. By wisely designing gene barcode combination, we can scale this experiment in a scale of whole transcriptome. This is just one kind of solution. You may design your solution with newly developed molecular biology technology. The second question is a relationship between compressive sensing and deep learning as my model presented. Deep learning model are, especially the autoencoder model, is considered as black box machine learning model. But it is well known. It is now become obvious that if we well design architecture, input-output relationship, then the behavior of deep autoencoder model can be explained by combination of transform and back transform as we saw in the presentation. The design condition is called deep convolutional framework, and you can find the detail of this kind of framework in the paper provided by this URL. And thank you for your attention.